I'd like to welcome everyone to the Diatom Web Academy. We started this about a year ago as part of the Taxonomic Certification Committee's effort to expand training for taxonomists and for the certification program. And with the pandemic, we had everything in place really to be pre-adapted and take off with maintaining connections with our friends and colleagues. And so we've had these sessions nearly every 2 weeks this year. So I think it's quite nice to be able to connect with all of you. And our speaker in 2 weeks will be David Williams from the Natural History Museum, London, who will be speaking on diatom classification. And today we have Dr. Maria Kahlert joining us from the Swedish University of Agricultural Sciences. Marina obtained her training in limnology from Uppsala University and the University of Cologne, where she focused on freshwater ecology. Her research focus is on benthic algae in freshwaters. And I think everybody will be delighted to hear that Maria attended the University of Michigan Biological Station, where she took Rex Lau's class on freshwater algae, like many of the people in this audience. So with that, I would like to turn it over to Maria.
Thank you. Do you hear me, everybody? It's okay? Okay. Yeah, I just start off and then let's see where we will end. I'm happy that Sarah asked me to present the work that we're doing here in Europe about the inter-calibration harmonization of diatoms in the Nordic-Baltic Network. And so my talk is going to be about the harmonization of diatom data. Yes, as Sarah said, I'm working at the Department of Aquatic Sciences and Assessment at the Swedish University of Agricultural Sciences. And here is Sweden. And my department is responsible for the national monitoring. We're collecting data and do status assessments for national environmental goals in Sweden. We work with the European Water Framework Directive and other European directives. We are also working or involved in international monitoring, for example, the Circumpolar Biodiversity Monitoring Plan from the Arctic Council. So, and a little bit about me here in this department, what I'm doing and why I'm doing it. So I work here as a senior researcher. My background is in limnology and zoology, and my research is focused on benthic algae. And I'm interested in their use as bioindicators because that's what my department is working on. But also I find it very interesting which environmental factors are steering their diversity, their biomass, and their nutrient content, and basically who is there and why. And yes, I learned my benthic algae from Rex Lau, and I'm still very happy about this course. And I found it really nice over there. Yeah, and why I'm doing it. I think as Jan Stevenson put it very nicely, human activities alter ecological systems in ways that greatly affect algae, and algae are excellent indicators of ecosystem change. So, so you can say for the sustainable, I'm working for the to fulfill the sustainable development goals. I mean, clean water and sanitation, life on land, which is including biodiversity in freshwater. So basically, I'm hopefully, at least I'm thinking I'm working my dream or doing my dream about saving the world, like maybe a lot of us we're starting off with. And so a bit, I'm hoping I can do that with my work, developing bioindicators. And I came into this harmonization work when I was starting with the European Water Framework Directive. there was kinds of groups called the GIG groups, geographical intercalibration groups, who had the objective to harmonize methods and boundaries for ecological status class, so that all the European countries would do their status classification in the same way. And I was involved in that with Martin Kelly and the Diatom work. And we were focusing on data that we had from microscopy and the indices and the ecological status classes we had from them. But actually, what I thought we were doing, that we would look down the microscopes, we never did. We never got the funding to really study and unravel national identification conventions in workshops. So actually, up to today, there's no European harmonization of the direct diatom identification. There's a harmonization of other things, like we have standards for sampling and analysis. We had in the first phase a comparison of the results. We had the harmonization of indices and ecological status boundaries. And one insight I got was that the most important factor of variation in diatom community on species level between countries was country, which was of course highlighting the problem that we have national identification conventions. Every country has their own solutions to problems. There were parallel projects to this work. We were comparing sample counting protocols. We were compilation all the intercalibration and harmonization work that is ongoing in different countries. We have started to analyze the effect of identification conventions. We also had some recommendation, but actually no real agreement of identification. So we know what we are doing differently in many cases, but we don't have a common solution to how to solve it. The problem is that even experienced diatomists, as you all know, do not necessarily end up with the same taxa list having the same sample. Don't look at the species names here or think about it. This is just an example of the same sample done by experienced diatomists, which end up with different names in different colors shown here. But there is no obvious analyst, what I found. There is nobody who is bad or something, but there is really a high variation within one sample. So these are different samples you see we're clustering together, but still, why is all this variation? And I was presenting this at the meeting in Luxembourg and hoping that the diatom experts collected there would join me on the way to make this better, to agree better, to get better harmonized taxa lists. What I got back was this: do your homework. I was told, standing there, that all these differences here, were due to the fact that people were not able to identify correctly. And I don't believe that. And that was why I started to think over the question, like, why do even experienced analysts get so different taxa lists? I don't think they are bad or we are bad. I think that is the problem. Original descriptions are sometimes a bit problematic. This leads to local identification conventions. So this is another example we have here, where you can see that the different colors of different people of the same sample are showing different species for the same, or different names for the same species. And the problem, of course, is when you work with bioindicators that the different species have different ecologies. So Fragillaria Forscheria is a tolerant species, whereas many, like of the other taxa here, are rather sensitive. So I think the solution is that we need more harmonization of diatom identification. So that's why I started to work with this. There's still there is some discrepancy, however, if we need this work at all. Do we really have to put energy on getting a better fine resolved taxonomy, getting more harmonized? Does it really matter? And this, I have to explain a bit about how is the diatom method in Sweden today, like the sampling with the toothbrush and working with the microscope. Of course, not of course, but we are using an index where in our case, all diatom species are included. We're using the relative abundance, and each species has an indicator value. Sensitivity value, which is showing the impact of a stress, and the tolerance value, which is weighting the certain species because some have a broader tolerance than others. And our used main index is the IPS, the index. My French is not so very good. It's a French index, which indicates eutrophication and organic pollution. And we classify from 1 to 5, high to good, by comparing with an expected value. And the European index used for count comparisons is an artificial one, which is called ICM, and combines this IPS with another diatom index, the trophy index from Austria. So, if you use this ICM index and compare it, the original one, and compare it with an ICM index based on a streamlined diatom taxa list, which means you don't have to find resolved taxonomy anymore, but you merge all the complicated taxa together. So does it really matter if you have to find resolution? And Kelly showed, and I was part of it, in these cases, it doesn't matter actually. You can have quite gross diatom groups here, and they still show exactly the same picture like the original ICM. So probably we don't have, so I got the argument, you don't have to put any energy in harmonizing diatom identification because it doesn't matter for an index. So people say, no, it's not necessary. But I think yes, because I show you this example here. There are exceptions, but these exceptions can be very important in bioindication. If you have a non-harmonized taxonomist, you can get differences in diatom indices. There are outliers, which can be very different and show differences in ecological status. For example, this is one of our NorBAF intercalibrations, which we also did with the European people. And here's the IPS, the index that we are using. And this was the one sample, and this was the range of IPS values that we got from all participants, from actually almost moderate status to high status, which is not normal. So, and this was based on the fact that some people separated Achnanthidium minutissimum, not separate, but they found not only Achnanthidium minutissimum, this one, but they also found Achnanthidium catenatum. And Achnanthidium catenatum has quite a low sensitivity value where you get an IPS value which is low, which means that the water quality the water status is not so good. If you don't count Achnanthidium catenatum because you don't recognize it or because you think, yeah, you don't see it, and then you get quite high values. So if you average those two, you end up here. The solution of Kelly and others to streamline these two taxa, because they are complicated to separate. Not all people see them, so we can just merge them, so they would be streamlined into one group. But then you end up in this index. Yeah, there is very low variation, I agree on that, but it is the wrong index because it shows high quality, whereas the real water quality is actually lower. So I think it matters. We should put in some effort into harmonization, at least for important taxa, for taxa that really makes this difference. And not least is this important if you have an index which is not based on just sensitivity values, but actually on the taxa list, where you compare not only with expected index values, but where you compare with an expected community composition. So if you have this kind of indices, like which Finland, for example, has, you can get in 1 year, this is a similarity matrix only, so you see the closer together these points are, the more similar they are to each other. So you see those 2 points in 2007, the diatom community was put up here. But before all the years, it was down here for both. This is one side. So you can either say, okay, the water quality has changed a lot, or what you actually see here is that they have changed the laboratory. After they saw these results, they switched back. And this is just because of a non-harmonization of diatom identification. So I think it's very important we put in effort to harmonize identification. And that's why I started the Nordic-Baltic Network for Benthic Algae in Freshwater, NorBAF. And I will just explain a bit who we are. A cooperation of people working with freshwater benthic algae in Scandinavia, including Iceland and countries around the Baltic Sea. We want to, most of all, establish and develop contacts between everybody working with freshwater benthic algae, educational institutions, research institutions, non-governmental organizations in the field of education and lifelong learning. This is very important. We are open to anybody who shares our interests. We are cooperating with other phycological networks. We started 2005. We got some help with from NordPlus Neighbor, which was one of the Nordic Council of Ministers 5 mobility and network programs, and also from University of Uppsala. Our focus is on diatoms, not because we don't like the other algae, but because here's the money, at least for the moment. Yeah, and we don't get any money anymore. So NorBAF is organized as informal cooperation of the participants. That is one of our meetings, or that was one of our meetings. So that's where we have people from. On my email list, there are 80 people from 13 countries. There's also some interest from countries that are not actually around the Baltic Sea, but they want to participate as well. And researchers and technical staff from universities. We have consultants, both from small and big consultancies, staff from government agencies and regional management boards. And we also have a few private people. And we have different interests. For example, the consultants, or many consultants, need a certificate of knowledge that they know diatom identification. And in many countries, diatoms actually are counted by private companies for routine environmental monitoring, but not in all countries. Both consultants and others want to get taxonomical updates. They want updates on ecological research. They want to have training in identification. And some of us need to have it documented, so they need to certificate that they actually do it. And everybody agrees that we want to exchange because we're not so many in the Nordic countries. And most of all, we are convinced that it is important to harmonize diatom identification among the Nordic-Baltic diatomists. And yes, globally would be nice, but we need to start somewhere in order to improve comparisons between different studies. Not least because diatom monitoring is now often a standard in aquatic assessments, following the Water Framework Directive, and also because diatom diversity is used in many research studies. So this is especially the diatom structure. And so it's important that we at least make people aware that it is important to put some effort in the identification. Yeah, we are convinced that the practical exercise combined with the friendly discussion of the results is an effective way to find disagreements and to agree on solutions, which is something we do every 3rd to 4th year. We count slides, and then we have a workshop at the Normalma field station at Lake Ekin. This is the Uppsala University field station. And we have right now an inter-calibration ongoing with the workshop in November this year, virtual. And it's mainly me organizing. And the practical, how are we doing it? We count 2 to 6 slides, sometimes also preparing them. This year we had 3, also prepared all of them. There's an instruction letter which says that you must use the Swedish national method and a master taxon list. It's quite detailed, this instruction letter. There's an anonymous collection and an analysis of the results following Kelly, which says you should have at least one auditor and you compare a similarity of results. And we have 2 auditors. And we also calculate and compare diatom indices, which are the Swedish ones, because we're also using the Swedish method. In short, our method means that we have to identify the species level and use a master list. There's one exception: the Achnanthidium minutissimum complex is defined by the valve mean width. So we are not going into species here. We have three groups: small, thick, very thin, middle, and very thick. So we count at least 400 valves, and we also have a certain method we call count deformed valves to have a screening method for toxins, but I'm not going to talk about this here because my time is restricted. If you are interested, you can ask me later. Yes, and if the participants pass a certain threshold of similarity to the auditors, for all samples, they get a certificate and count as approved by NorBAF. In some countries, the authorizing institution are supporting our activities, and in Sweden, the certificate is actually officially used as one important documentation of diatom knowledge. And there are also the official institutions are in Sweden and in Finland informed about our activities, and they know who is participating. And even if we have anonymous results, they can ask the participants for their results. So they can ask if they were approved or not. Yes, and what are we doing? Just in practice, how are we doing it? So we have counted the results. Now we are at the workshop. And the first thing is we make a species list with the most dominant species. And then we go through all of them, discuss them, how they look like. And so we agree at least on what is most dominant in a sample. And on that way, we are already going through some of the taxa that you easily can mismatch or misidentify or is problematic. And then we put these nice colored graphs, and then we go through the main problems one by one. For example, one of the most dominant ones. So I show which of the taxa are in the sample, which are not in the sample, and everybody can, in this way, we have numbers, so you don't see the names, but everybody can in this way say how much they actually deviate from others and from the auditors. If this is the auditor's mean, this person can see, oh, I have a problem because my colors look very different. Yes, and then we go through all of them. So, and then we have two kinds of problems. We have beginners problems, which we in this year will discuss first on a pre-workshop because the advanced people told me that they can't hear it anymore. So participants do not follow the instructions. Anybody who has done this, organized these comparisons probably knows what I'm talking about. And then you have also often cross-contamination of samples, often by using cooking in open beakers. Don't do that. Then usually one problem is that people count tabularia flugulosa girdle bands. They always do, even if you have instructions, they shouldn't. And then one important thing that I think everybody should now and then check: the overlooking of small taxa. Because usually this leads to a lack or severe underrepresentation of small Navicola sensulato. And I'm not saying this is all Navicola, you know, all these Selaphora, Eolimna, and so on. Small taxa looking like Navicola in polluted sites, very frequent. And you get, if you overlook them, an overestimation of the ecological status class, a too good diatom index, because they usually occur in polluted sites. You see that nicely here. This is our index. This is good quality. This is bad quality or moderate quality. This is really bad. Here are our auditors. You see have 25–30% of small Navicola sensulato. And if you overlook them, if you have 0 or very few, you get a much better index. And this is actually, in this case, linear. So very important thing. And then I show everybody their deviation of the IPS and their similarity to the auditors. So if you would have a 1, you would have exactly the same taxa and the same counts, which you never have. like the auditors mean. And if you have less similarity, you deviate more and more. And at the same time, usually the deviation of the IPS also increases, which is a very plastic graph showing that, okay, we had 4 samples here and number 8 was deviating in all of them with the IPS and also with the similarity. So this person clearly sees directly, oh, I have a problem. And if you put away the beginners, if you take, we have a cut here with 0.6. So everybody is has a higher BC, break-Curtis similarity, is approved, and all these people are not. You really get down the IPS deviation. You don't catch all problems, but you catch the worst problems, which I can show here. If you put all participants and the index, you had the 4 samples here in our study, you get quite high deviations. But if you take away those that have less than 60% similarity, you get a much better, or much less variation. The green bar is the allowed variation we have for this index in Sweden. Still, you get some problems. And that's so the next thing, that is we are discussing what is actually still causing the high variation. This is the more advanced problems. So we go into the identification harmonization, not the beginner's problems. Where to start? We never have time for everything. So we first look at which taxa cause the trouble in the diatom index calculations. So which are still causing a high variation, which taxa are those making the high variation. And this is what we found so far. It's always Acnanthidium minutissimum complex, like this catenatum that I have shown, which is not minutissimum, but this is part of our complex here. We have a Gomphonemma parvulum excellissimum, which is a big problem. The Fragillari cappuccina complex, Nichia Dismal novicula, which some of them have not low sensitivity values. The overlooking of typical Nordic taxa, which are not in the common books, like the European Süßwasser flora, is also a big problem, because they're usually having oligotrophic, or they occur in oligotrophic conditions. If you overlook them or you identify them for something more polluted, you get the problem in the index. We have quite a lot, Psammotidium abundans, Pusilum, Acnanthidium krieri, for example. And also the same problem here, people mistake oligotrophic navicula, bigger ones, for common eutrophic ones, because they are in the books and the others are not in the books, or are not in the common books, or are less common identified usually. And then we have a second question, which taxa are identified very differently by the participants, actually often not causing IPS deviations, but causing problem to identify them. And people make notes that they want to discuss them. For example, we put in this similarity graph again, but this time more detailed. So you see the participant here. And then you see again which taxa are driving the participant to the different directions. And then we really go into these detail. We have removed the beginners. Now there's the other people left. So they see, okay, in this case, this Gomphonemma is only or mainly identified by participant number 9. And, oh, sorry. And this Gomphonemma is only mainly identified by participant number 10, and that's actually what is one of the reasons why they are driving apart in their analysis. And then we go through these 2 taxa, or if there are more, we go through all of them and really check the differences of these taxa explicitly, because they are causing the problem here. So that's what we do for all the samples during the few days we are meeting. For example, in 2009, just an example what we have done, we have worked with Fragillari a lot. where you can see the people having the red taxa, they identified Faucheria, the yellow was a Capucina group, and somebody had Capucina sensu stricto, and that was this taxon, which I have heard from taxonomists is Fragillaria pectinalis, and maybe others as well. The same sample was analyzed in the European harmonization, which I showed earlier, the one I showed in Luxembourg. Then we got even more names than those. And it got even worse when we compared the identification on global scale, when we had the North American taxonomists or diatomists included, the list was even longer. So this is a problem. This complex really is a problem. So we want to agree on it. We want to agree on how to identify these taxa, not because we are very good taxonomists, but we want to have an easy agreement which is possible to use in routine identification. So what we are doing, I'm not telling you that these names are the most updated ones, just an example. So for example, long thin Fragillaria was a problem. How to call different things. We show all the pictures and then we discuss how they look like, where are the differences. For example, this one has like kind of shoulders, this one has not, even if both have a belly. So we agreed, okay, they are not the same. This was called Capucina sensulato. This was count subspecies rumpens. And we agreed, okay, it's not the same than rampens. And we discussed a lot now. These two have a new name. This one is pararumpens. This one is nevadensis. And then we make this table. Like, okay, we know now this table is much longer and it's not up to date because, yeah, I'll show you. But we don't make own conventions if there already are good thresholds. So we use everything which is available, put it this kind of these tables, make characteristics, like discuss, okay, we think we can use these characteristics. For example, for gracilis, we think stria and sides are parallel. For tenor, for example, stria alternate. And we also look what the width is and so on. So we have suggestions how to solve identification problems, and we try to follow them in our work and also in the next intercalibration. But, so of course, how to deal with updates in taxonomy. Shall we use all the new names because you know that the Fragillaria list is much longer nowadays? Or should we wait for some approval of the Diatom community? How to deal with long time series where we have used old names? How to deal with agreed identificate conventions that we have used for some years? Shall we just make new ones? Do we even agree with new names? And are they making a diatomist's life easier or more complicated? And are they relevant even for environmental assessment? People come like these workshops, for example, by Bart and others. And I read it through and I learned a lot of even more new names, which are complicated to separate from each other. And I'm also convinced that many things are not possible to identify by light microscopy. And how do we even deal with DNA barcode species, which are cryptic ones, which you can't really identify, at least not by light microscopy. How to deal with other agreements of diatom identification. There's a polymarine diatom workshop. There is your North American side. There's a lot of different things that are already agreements, not necessarily the same than we have. And how shall we continue? But I'm still convinced that we need more harmonization. And most of all, I'm convinced we need more communication. We need work combining morphology, molecular and ecological research. We need more constructive communication among scientists and also with the environmental assessment. Agreements on practical solutions for identification and communication, not least to non-academic analysts, like the user diatomist biomonitoring. I mean, the researchers can't just work for their own and expecting that the routine user just accepts everything and also understands how to separate everything, or if something cannot be separated. I think we need agreements there. And we also need to communicate why this is important, because it is affecting the ecological status classes. So this is a lot of work. So we need persistence and patience and a long breath, but we certainly also need shorter term solutions for usage. And yeah, that's what I wanted to tell you. That was the thing I have prepared. I'm not sure if this was what you expected to hear, but I'm happy to answer any question.
Thank you so much, Maria. It's an excellent presentation. Thank you for sharing your research. And I can imagine that the effort, you've put in so much effort into, you know, organizing these inter-calibration meetings, and there's been a lot of communication that you've fostered. So it's really impressive to see that. I'm going to just check on the chat here, and I think people will start typing in their questions. But to get us started, I was curious about, I think near the beginning of your presentation, you mentioned there was a streamlined taxa list and a Swedish master taxon list. How were some of the taxa groups determined? Like, you know, the difficult groups, you know, was it mainly expert opinion on which taxa to combine to have more of a streamlined list?
I have, let's see. I'm not sure if I'm sharing the screen now, maybe not.
Yeah, I can see your screen.
Yeah, if you're, I must admit this is a while ago, but there's two publications where we give some more details. If you're interested in the lists, actually they are in here. But mainly what we did was putting all taxa together that we found people have identified differently. So we made a group saying, okay, people identify this, probably it's the same taxon, identify it differently. And so we put everything into one group. There was not an agreement. It was necessary to work in the European intercalibration groups because otherwise it wouldn't have been. Otherwise, we would just have found out that the countries are differing, and we worked with the ecology and pressure relationships. So we needed to see, okay, which taxa actually are the same. So it was more a working item. It was no agreement at all. Yeah, but you can easily see the groups which people are identify differently there. I don't have everything in my head, but please. And I don't. Yeah, as I showed you, I'm not agreeing in many of them. I'm agreeing that we for this work had to put them together, but I don't think you should use them for routine analysis.
Okay. Related to that, Mark asks, does NorBAF have any published reference images for the master taxa list?
No. There is also no NorBAF master taxa lists because we are an informal network, and the things we are doing is even if people want to have a certificate, we are still having it very informally, everything. So the master taxa list is a Swedish master taxa list, and there are no images because I try to say that we wouldn't need that, but and I like very much what Sarah is doing and what you are doing in North America with the Diatom Web, with the North American Diatoms, but we don't have any resources. We tried to get resources to make this for Europe with Martin together, but we never got funded.
I did think that the table you showed with the detailed, you know, characteristics, that seemed really helpful. Even without images. Images would be even better, I think.
Yes.
Mm-hmm.
Yes, I agree. That at least this one is published, but this is out of date already. So we have we said that we this year would try to to get a new publication out with the recent recommendations, because we haven't managed in the last years. Everything, we have agreements, but we never published them properly. So we will try this year. We'll see how far we how far we come. Yeah.
Okay, we are getting more questions here. Ewan asks, has anybody actually shown that processing the samples in beakers has resulted in cross-contamination, like the open beakers?
Published? Yeah, I don't know. I haven't searched for it, but you can easily, I can show that from from the results of the people, but I have never seen it published.
Okay.
Maybe it's also not that the diatoms are bubbling out. Maybe it's like it's open, so people are contaminating it by working with these things without logs. So I don't know.
Yes. Is it some of the common beginner problems that you saw?
Yes.
Okay, next question here. Sarah asks, Maria, you showed a graph that indicates that you use the Bray-Curtis similarity as 60% as the acceptable level of similarity. And of course, there's also natural variation in each sample or the slide or the transects. Have you tried testing people by using LM images, that is to evaluate participants' abilities?
No, we never did that because there's simply no yeah, I mean, we are an informal network, and I can't say that I'm the big teacher, or I must be very careful to say something like that. So that's why we relate on a published intercalibration or a published harmonization method, which is Kelly's from 2001, where you can say, okay, he did it, so we do it the same way. So we rather do it the same way and then discuss during the workshops to make, I mean, when we show these slides, where people see directly that they don't have any yellow color here, we hope it's pedagogical enough that they see that they need to improve their identification. But we never had like tests showing an image and then people should answer on that. Because most of all, my auditors too, they are very different, and they actually used to fight in front of the public which taxon it is, if they are both there. And nowadays I'm enjoying it, but it's showing very clearly that there is, it is not so easy to identify than I thought from the beginning. I think we should go for this table saying for routine identification, we use these methods. It's not necessarily true that everything can be put into these tables, but it helps to say, okay, I put that name because in the table it's ending up there.
Yeah. So for these tables and recommendations for intercalibration, a lot of these are already published, right? You have in your previous publications, you know, some of the taxa that were combined, recommended to be combined.
Yes.
But then you also mentioned that, you know, it's already outdated.
Yes.
So how do you have recommendations for, like, you know, someone who's trying to, you know, look at these references and figuring out what to do with their own samples?
No, or yes. You see, that's exactly the problem. How to move forward. No, what we do right now, we try to use our recent recommendation. We have a homepage like the NorBAF net, where we publish everything, even if it's not published. So all our presentations are published there, and then we use. It's more or less a work in progress. So we use the tables that we have agreed on the last meeting. But it's often that they're not all taxa are included and so on. But we try to use the most recent one. So we have that one still, but it is actually built on here. And we need to update it. But there is more work than we have. I mean, it's all voluntary work. Nobody gets paid for this. And yeah.
Yeah. Marina asks, what happens to the slides that are made as part of these European monitoring programs? Are they deposited in museums? Are the samples saved?
It was just one, which was a voluntary one, which I organized. People have the slides at home. I have them at home. They are not public available.
What about the bigger monitoring programs for the Water Framework Directive?
If they are harmonized, no.
Like what happens to the actual microscope slides? Do they stay in people's laboratories? Are they archived in herbariums?
This is Europe. Each country has their own rules. So I don't even know how the other countries are doing. In Sweden, the slides are ending up in not in a museum, but in our archive at the university, because we are the national we have the national diatom counting, so they are here in the basement. But there is no museum. We don't even have diatom slides regularly archived in the museum, because in our national museum there is no microbiologist or expert of microorganisms. So I don't know how this is in other countries, but yeah, that is really unfortunate. It's not easy to get hold of these slides.
Mm-hmm. Yeah,
and there's certainly not pan or European agreement on that.
Yeah. So once you go through this intercalibration, you know, were you able to test whether all of that effort, like meeting together, did it help, you know, the analyses later on, the subsequent analyses? Did the participants learn and change their analytical techniques?
Yes. Well, one of the nice things, and why I'm still continuing, is that you can see people who have been here ending up here the next year, or the next time. Already the next time they're doing it. That is the really nice thing. So it's not because they have learned necessarily, but because they have, because they can be very experienced diatomists, with having their own kind of work with problematic groups, calling them names that others don't use. But if we agree on using the same methods and the same thresholds and so on, the same tables, then we end up here, which is very clear. I don't have a publication on that. Oh yes, I have actually one of the harmonization publication that 2009 that I published is showing that harmonized people are closer together than somebody who is just coming new into the group. Mm-hmm. So yes, it is. We're getting better also in the deformations counting. If you do it the second time, usually we are closer together. But we still have these outliers when there is a new sample with unfamiliar taxa. Then again, we are all over the place, and then we're discussing, but we get more and more taxa. On the other hand, also the taxonomy is updating continuously. So.
Yeah. So what happens, you know, after you come to more agreement on these taxa lists, you know, what happens to the older data that were identified with, you know, everyone else's, you know, individual way of identifying?
That is still, I don't know how to solve this. This is still, we don't have any routine how to deal with this. The only thing is that you have to go through the taxa list from old samples and see, okay, this is something we call now this. And this is what I have done with people from Finland coming with old samples, and then we sit together and go through each taxon. Because okay, if somebody has a simple way to do this, I'm open for any any suggestions. But yeah, you can't compare the old and the new taxa lists just like this. You get an impact of this method. Yes.
Yeah, it's not easy.
No.
I was also curious, you mentioned that Agnanthidium minutissimum complex group is identified by the mean width. But what about all of those valves that are in girdle view? How do you, you know, you can't get the valve width there.
The main point is that we don't have to count them all anymore, or to identify them all of them. So we just measure 10 to 20 valves, and the rest is just counted. Girdle or valve view doesn't matter. So just 10 to 20 are measured. We calculate an average, and all of them end up in one group, which makes things very simple.
Yeah, it's very practical.
Yeah, and the main thing is that the fat ones usually indicate eutrophication pollution, and the thin ones, the very thin ones, usually just are in the mountains, and the middle ones are everywhere else, and that's what we want to indicate. So we don't. It has been shown that it is so complicated to get a harmonized taxa list on Agnanthidium complex. We don't have any recommendations there, so we rather go further with.
Well, I'm not seeing questions here yet. Maybe people are still thinking of some, and we do have a few more minutes. I thought it was really interesting in your slides that you kind of saved it for time purposes about the counting deformed valves. Would you like to share a little bit about that?
I can try. Let's see. I don't know if you see that.
Yes, we can see.
So this is we're just using it for screening. So we count the teratological valves, and we have two different categories: the abnormal valve outline, which you can see here, like if this is normal valve, and this is slightly deformed, and this is severely deformed, and we have this for the most typical taxa. Or frequent taxa. I don't know. We have pictures for that in a compendium, so you know what to call normal, to call weak abnormal, and strong abnormal. Then we have the abnormal valve ornamentation, like the stria are strange or the raphe is strange or something. And so we count them, and then we have the and we sum up both of the categories, and then the percentage of the deformed valves are classified in these 5 classes, like less than 1% of all valves is none or very low frequency, and higher than 8% is very high, and at I think at 2%. There is a warning signal coming up in the system saying, okay, if it's more than 2%, you should go and measure metals and pesticides and maybe other toxins in your water, because there's something, probably something strange. It's not necessarily so, but the probability is quite high. And if you see the harmonization here for 2013, you see there's, in one sample, it was a low frequency in both, it was the auditors had low frequency in both. There's quite a lot of variation between people, but if you go the 3 years, the last intercalibration we did, it was just 2 outliers. Everybody else was nicely on the line, and we had also about the same amount of weak and strong deformations. There was 2 outliers, and those 2 people were new to the group, so they never had done this before, and the others all had participated the year before. So I think this is nice that you can get down to this level of agreement. So, and then this is used as a screening method in Sweden. In Sweden only, actually. I don't know of any other country who is using this.
So Sarah says she's familiar with these teratological valves being result of pollution by metals. Is that the same in Sweden, or are there other causes of deformity?
Metals are the most probable causing this, but I also found that certain pesticides are causing this. So we have some agricultural streams where there's a heavy use of pesticides, and there you can get these deformations as well, but not for all the pesticides, but for certain, and I don't have them in my head right now, of course, but for certain ones you can get this as well without metals. But you can also have an impact of metals, or it's more probable that you have an impact of metals, yes.
Sarah also asks, you said these results are all based on 400 valve counts, is that correct?
Yeah, 400 or more. We don't have a strict upper limit. Or we don't have an upper limit. If you want, you can count 2,000, but you need to count 400. And then we recalculate everything on relative abundance to make it comparable.
Are there any issues with, sampling effort? Do you think that might affect the values, your outputs?
Yes, there is. If you don't have really time and put this in the last minute into sending, and haven't measured your actinidium minutissimum well, and it's on the threshold. If you have just measured 4, yes, there is a low sampling effort, and then you can end up in the wrong group. And this is actually something that has happened several times. People don't take it really seriously sometimes to measure, and then but we usually the differences in counts are not so high. People count everything from 400 to 450 taxa, and I haven't seen any issues regarding a similarity or regarding index in this relatively small span. People don't count 600 valves if they don't have to.
So how do you know if you don't have to? Or is it just like a...
People don't have time.
Yeah.
They need to count 400 because that is the rule, and they don't count more. You don't, in routine analysis, you don't, at least in Sweden, you don't put in more effort. You just by chance count more than you have to. But people pay for this exercise at least so much that we can at least organize it, and then they usually don't get paid to do it. Some people do, but many analysts they actually do it on their free time, and they don't count more.
Do you know if that 400 number, was there any research to say, that's enough? Sufficient number of valves to count?
No, I was looking at Sarah at Meredith's, Thury's work and Sarah's work about this, how much is enough and so on. But the thing is that most of the European countries are not looking into richness at all. They just calculate diatom indices, and they are very very robust against few or many taxa. You still get the same index value. The rare taxa are not counting very much in this kind of system. If you have a structure-based index where you need taxa list, then it matters, of course. And then of course the similarity also gets big issues. But usually it's not the breaker. The similarity is focusing on the dominant taxa. So those are the ones giving the problem the same as in the indices. If you would use another one, you would probably get sampling effort issues. But richness is not so important for our analysis. But yes, I'm sure. But there's no reason why we count 4 in it, I can tell you, because it's just a habit. tradition. 400 was the agreement.
Okay, we are at the top of the hour. I want to thank Maria again for sharing her experience and insights on this topic. I hope everyone stays safe and joins us again next time at the Diatom Web Academy. Thanks, everybody.
Thank you.
Thank you, Maria. Thank you, everyone.